This is the fifth section of the recorded lectures on viruses. We talked about animal viruses a little bit, and then we talked about, and we are now talking about plant viruses. Down here at the bottom, plant viruses can be transmitted by a variety of vectors. I'm not sure. I'm trying to get my pen. Give me a second. Okay. I'm not sure if I've told you exactly what a vector is, but a vector can be, it's just anything that carries the virus. So through contact with an infected plant's sap. So there's a plant that's infected and it's releasing sap, kind of like in humans, maybe pus, or blood, body fluids. So the vector is the sap, okay? Vector is just the mode of transmission. It's just the thing that transmits the virus to another plant. And so a vector can also be an insect, a nematode or a roundworm, and pollen, because these are all living things that can come. Well, pollen's not really a living thing, but it's contains a sperm of a living thing. But anyway, all of these can act as vectors because they can come in contact with the plant sap and then they can transmit the virus that way to another plant. So when plant viruses are transferred from plant to plant, that is horizontal transmission. It is very important that you know this because I know it's on your quiz. Horizontal transmission is like if an insect gets a virus from tree sap, like from a maple tree sap, for example, and that insect goes and lands on another maple tree that has an opening because it's got damage, and through that damage, the virus enters, so that would be horizontal transmission. And then when a parent transmits the virus to the offspring, in other words, when it comes from the seed of the offspring, then it's vertical transmission. So the parent plant basically transmits the virus through the seed or through the sperm or the egg or the pollen or the ovum. But anyway, it's inherited from a parent plant, and it's not transmitted by a vector. Some of the symptoms of plant viral infection are hyperplasia. Hyper means higher, so that's going to be extra growth, and that can appear as galls on tree limbs. The galls look like big swollen balls. They're just round, big swollen areas on the branch. You can always Google these if you want to see what they look like. Just Google gall and images, and it shows you. There's no images in the slides, but you can Google them in your own time. But galls are basically excess growth or tumors on the plant. Hypoplasia means that the plant is thinning. Maybe it has yellow splotches on the leaves, and it's actually withering and getting smaller. Necrosis is when areas of the plant are dead, and we've all seen that blackened area on stems, leaves, and fruit. Abnormal growth patterns, malformed stems, leaves, or fruit, and then discoloration, different colors in different parts of the plants. Okay, here we're seeing a vaccine is being given for prevention of a virus. Vaccines are usually, COVID is different, but vaccines are usually made of live viruses that have been weakened. That word is attenuated, or killed viruses. That means viruses that cannot cause disease. That So let's talk about how that works. Live viral vaccines are designed in the laboratory to cause few symptoms. And most of the time what happens is to attenuate a live vaccine so that you can give a person live virus and encourage their immune system to develop immunity in case the real virus, the dangerous virus attacks them. What you do is weaken the virus, but you weaken it by growing it in tissues that it's not accustomed to, like maybe a human virus, but you grow it in a chicken egg, or at a temperature that's different from what it's accustomed to. If it's a human virus that's normally accustomed to 98.6 degrees Fahrenheit, then you grow it at a colder or a warmer temperature. And that will weaken the virus. That will cause the virus to mutate to a weakened form. The problem that this can cause is anyone who's had a flu vaccine and gotten the flu from it had that happen because of a back mutation. That means the weakened virus mutated back into the stronger, more dangerous form of the virus. And that is a very small risk that can happen when you get a live vaccine. Some vaccines always have to be updated, I guess you could say, modified. The flu virus has to be modified every year to account for mutated strains of influenza. So viruses with high mutation rates, the vaccines just have to be modified to treat the new version of the vaccine. Now, sometimes vaccines are used as treatment, and you may have heard about this with the one I can think of is rabies. If within early on of you getting bitten by the wild animal, that's usually how a person gets rabies, and it's very, very rare, but it does happen. You have to get a series of vaccinations, and what that does is boost your immunity, and hopefully in time for you to eradicate the virus from your body. So vaccines can be used as treatment in the case of rabies, for example. And then there are antiviral drugs. With the flu, you may have heard of one called Tamiflu. And what Tamiflu does is, or what any antiviral drug does, is interferes with some portion of the viral infection steps that we learned about. Tamiflu inhibits a viral enzyme called neuraminidase, or NA, in the influenza viral envelope. So it's an enzyme that's in the virus originally before it infects your cells. The neuraminidase cleaves the connection between the viral hemagglutinin in the viral envelope and glycoproteins on the host cell surface. Inhibition of NA prevents the virus from detaching from the host cell. So what it does is it doesn't kill the virus. There's no such thing. It doesn't stop the virus from infecting that host cell, but it stops the virus from moving on and infecting more host cells. I've had Tamiflu within hours of getting flu symptoms, and literally within 2 or 3 hours felt great. If you get it at the right time during your infection, an antiviral drug can be incredibly effective. This is showing you some antiviral drugs against HIV, and this is what makes a person living with HIV is not a death sentence anymore. There are anti-HIV drugs that give the person extended life of years and years and years and years. The person can actually live so long that they don't even pass away from HIV. They get hit by a car or some other way they die, but the HIV does not develop into AIDS, and it does not kill them. So HIV is a RNA reverse transcriptase virus. And so this is showing you the steps of HIV infection. First, HIV fuses to the host cell with the glycoproteins and the CD4 receptor. So this host cell is a T cell, which is an immune system cell, and it's a helper T cell, which is basically a helper T cell activates the other cells of the immune system. So if you can kill all the helper T cells, you can shut down a person's immune system. That's why HIV is such a bad virus to get. So after it fuses to the host cell, the HIV RNA, reverse transcriptase, and integrase, which is another enzyme, and other viral proteins enter the host cell. And then the viral DNA is formed by reverse transcription. The reverse transcriptase came from the virus and it was injected into the host. Viral DNA is transported across the nucleus, and now you can see it integrating into the host DNA. And now that viral DNA is going to, any gene that it codes for is going to be produced. So the RNA that's found in the core of the virus is replicated, and that new viral RNA is used to make viral proteins. And then all of the components of the virus escape, and an immature HIV forms. And then outside of the host cell, I keep trying to find my little dot. Outside of the host cell, step 7, you have a mature virus, so it matures outside of the host cell. Okay, so any antiviral drug is going to have to inhibit some phase of this cycle. It's going to have to inhibit some phase of this cycle so that you don't get a mature virion as a result, and that can infect another host cell. Okay, now we're going to talk about prions and viroids. Prions are proteins. They're only proteins. They do not contain nucleic acid. Gosh, come on, pen. They are infectious agents that consist of only protein. And a lot of them will produce, their infection symptoms are neurological. Mad cow disease is a prion disease. Here we go. Hold on. So prions are infectious agents that consist of only protein. And they are mutated protein. And this goes over the cycle of how prions work. But basically, they do cause diseases like mad cow disease. They cause symptoms in nervous tissue. Viroids are only plant pathogens, and viroids are only single-stranded RNA. It's just a single-stranded RNA. That's it. They infect plants, and they cause plant diseases. These potatoes have been infected by a viroid. They only infect plants. That's very important for you to know. Okay, and then we have a practice question. Why are multiple antiviral drugs used together to treat HIV? It's kind of like when we use a cocktail of antibiotics to treat a bacterial infection. It's in the hope that if one drug doesn't work, that the others will. Okay, and then there are several questions here. I don't know if we'll have time to answer them, but I'm not going to do another recording. How were viruses discovered? Remember, that was through a special filtration process, the Chamberlain-Pasteur filter. What theories do scientists have about their evolution? Make sure you review the devolution, escapist, and self-replication hypotheses. They're not theories. Viruses can be classified many different ways, but we use the Baltimore classification system, which consists of 7 groups, and I'm going to end there.